CAR T cells are T cells that we take from a patient who has cancer. They're the immune cells that are in the patient. We take them out, we re-engineer them so that they target a specific marker on the tumor cells, and then we give back those T cells to a patient to help their immune system fight the cancer. I'm Natasha Kekri. I'm a hematologist at the Ottawa Hospital, and I work in the bone marrow transplant program within the Division of Hematology. We've done this in certain leukemias and lymphomas where patients have not responded to any therapy. They haven't responded to chemotherapy or stem cell transplant in some situations. And yet we've seen with this therapy remarkable responses. We've seen anything from 50 to 80% response rates in these diseases where patients really had very few options. And that's why we're so interested in this, because getting this therapy in Canada has been incredibly difficult, and there is not yet a Canadian clinical trial program for CAR T cells or good access to the products that are now approved in the US. So we came together as a group, John Bell, Brad Nelson, Robert Holt, myself, and Dr. Atkins, and basically came together as a group as part of BioCanarex, which is a national funded program, to try to decide a way that we could bring this therapy into Canada. So because we're probably the only center right now in the country that has GMP grade virus manufacturing, that means that we can make the virus for clinical trial, not just for us to use in the lab. And that alone makes us sort of able to run a program like this, where we can make clinical grade virus, which is the part you need to code these T cells. That's the first thing. And in addition to that, we have Dr. Dean Ferguson and Dr. Manoj Lalu's team, who are also here in our epidemiology program, who really think about clinical trials differently than a lot of people do, and have allowed us to create an implementation strategy for building these clinical trials so that we have capacity ready to go, we have the right patients ready to go. We've thought about what barriers there might be in running the clinical trial so that things don't fall apart when they're going from bench to bedside, so that we have a real program in place before we even open the clinical trial. So in patients who have what we say is relapsed or refractory acute lymphoblastic leukemia, so these are patients who have either failed aggressive therapy like chemotherapy or stem cell transplant. Patients like that who have failed those therapies often have no other options. When we have made these CAR T cells for those patients, the responses that we've seen have been anywhere from 60 to 80%, depending on the clinical trial that you quote. And in a good handful of those patients, we've seen very durable responses, meaning that we've seen patients who are now getting closer to 5 years out from their therapy and still having no evidence of their leukemia. So we don't like using the word cure very lightly, but those are patients who we would deem to be cured of their underlying leukemia. In lymphoma, we've had similar results, again, with relapsed refractory disease, patients who have failed standard therapies. They haven't been as high as they've seen in leukemia, but again, these are patients who had very little chances of response to other therapies and have done quite well with CAR T cells. Our ideal plan is to provide the virus part, which is what we think is the most important part to making the T cells. That part will be provided by the Ottawa Hospital to any center in the country that would like to participate in our program, and we hope that we can build a very large network across Canada.